Scientists often need to use model organisms to do their research because it's not always ethical to use humans, and it's often not cost efficient or time efficient to work with humans either. So you might need to use a model organism. The first thing you should consider when picking a model organism would be, does it actually model the thing that I'm interested in studying? So if you're looking at cancer treatments, you might need to use higher organisms such as mice, whereas if you were looking at something simple like protein folding, not to say that it's totally simple, but you might be able to use a more simple organism such as bacteria to study protein folding inside of cells. What other things do you need to consider when picking model organisms? Well, the cost is important, so maintaining bacteria is often a lot cheaper than maintaining colonies of mice. How easy are they to work with? How many individuals can I grow in my lab space that I have? So you can grow hundreds of bacteria on one tiny petri plate, for example, but keeping something larger such as mice or birds or C. elegans, you can't have as many individuals in such a small space. Think about the maintenance. Is this something that someone in the lab needs to work with and take care of every day? Does it need to be kept in a special incubator with specific oxygen levels, or is it something you can just keep out at room temp in a small area and they can be maintained in the lab for long periods of time without having to worry about them, or you can stick them in the freezer and then bring them back out when you're ready to do another experiment. Other considerations would be safety. So you would always want to use the safest organism possible. So if you're going to work with a bacteria, pick one that is safe, that cannot get humans sick, rather than one that has the potential to make people sick. Think about what's already known about the organism. So you wouldn't just pick any random bacteria. You would want to pick one that we already have the genome sequenced if possible, that we know a lot of the function of proteins in it, that there's been a lot of literature on already. Also, sometimes generation time can be very important. If you're doing studies on aging or long-term experiments, then you want to use something with a shorter generation time so that you can complete your experiments without having to wait around for too long, right? And of course, ethics definitely comes into play. So you generally want to use the simplest organism that still models what you're interested in. So you would never use mice, for example, if you could accomplish your research goal using something lower, such as yeast. So those are just some of the considerations that go into picking a model organism. Common model organisms going from sort of more simple to more complex. So you've got bacteria such as E. coli is a very common one. You also have simple single-cell eukaryotes such as Saccharomyces cerevisiae, which is a yeast that we'll be working with. You have more complicated eukaryotes that are multicellular, such as C. elegans. We will also be working with C. elegans in our labs. And then you have more complicated eukaryotes such as fruit flies or frogs, and even plants such as Arabidopsis is a common model organism. And then for more complicated studies, you might even have to go up to higher organisms such as mice, rats, or chimpanzees.